student, Dr. Nikhil Sangith, who will be talking on, academic industry transition, arsenic, your expertise in a startup. As you know, all of you know that normally a student representative from the campus will introduce the webinar speaker. We have Ms. Rucha Kulkarni from ACTRA campus, who is a 5th year PhD student in life science in ACTRA campus. She will introduce the today's speaker. Before inviting her to introduce our today's webinar speaker, I just would like to tell all the students, if anyone is interested to give student webinar, then please write to us, write to me. We'll arrange as per the available schedule. Especially I will invite engineering students because we had life science webinar, physics webinar, chemistry webinar, but engineering webinar so far no student have given. Even former students who have just completed their PhD and doing postdoc somewhere else, if you know, that they also, if anybody is interested, they can also give such webinar for the benefit of our present students. So definitely I'll be waiting for this kind of response. I now call upon Ms. Rucha Kulkarni, who is the research scholar student representative from ACTRA, to introduce today's webinar speaker. Rucha, please.
Thank you, sir. A very good afternoon to everyone and to our distinguished speaker for the day, Dr. Nikhil Sangith, and his topic on which he'll speak on is academia industry transition, harnessing your expertise in a startup. We welcome you, sir. To begin with, he is currently working as a Chief Scientific Officer in Exact Diagnostic Private Limited. He is an experienced scientist with over 2 years of experience in industry and 4 years academic research experience after PhD. He has an excellent reputation for being innovative, resolving problems, and improving customer satisfaction. His specialization before PhD was in the subject of biotechnology. His PhD in life sciences was from ACTRA, Advanced Center for Treatment, Research and Education in Cancer. In the subject, his thesis subject was elucidating the structural basis of substrate recognition by the proteasomes, a global approach, under Dr. Venkatraman. Talking about his accomplishments, he has received a number of fellowships, which I'll be listing out. He has received a fellowship from the Department of Biotechnology, DBT Builder Research Scientist Fellowship, Tata Institute of Fundamental Research Postdoctoral Fellowship, Department of Science and Technology Senior Research Fellowship, Department of Biotechnology Senior Research Fellowship, Global Health Grant Fellowship from University of Massachusetts Medical School. He has received the DBT Junior Research Fellowship. Talking about the awards, he has received the Best Poster Award in the 79th Society of Biological Chemists of India, Indian Institute of Science, Bangalore. DBT JRF examination All India rank with 66. UGC CSIR NET Fellowship in Life Sciences. He was a gold medalist with first merit position in MSc Biotechnology and also a gold medalist with first merit position in BSc Biotechnology. After his PhD, he was a visiting faculty in Pillai College of Science and Commerce, Navi Mumbai. He was a postdoctoral research fellow in TIFR Mumbai, wherein he worked on identifying potential signaling hotspots in mycobacterial LGT that can potentially influence host-pathogen interaction. He was also a scientist at DBT Builder, Anna University, Chennai. He is a Chief Scientific Officer at Exact Diagnostic Private Limited, Chennai, where he is presently working. His talk today is focusing on startups and how to harness our expertise if we are interested in starting a new business. His talk also focuses on ideation of startups, its experiment design, its execution, validation of the idea, and the examples of success and failures that one can encounter during this journey of moving to and establishing oneself to a startup business. I request Dr. Nikhil Sangith to please begin with his talk for the day. Thank you.
Thank you, Rucha, for the introduction. Uh, Thank you very much. I would like to first thank HBNI for inviting me to this platform and share my experience, and also especially Professor Maiti, uh, who played a major role in inviting me to this platform. So the topic today, which I'm going to discuss, is academia industry transition and how we can use your expertise which you gain in PhD in a startup environment, which can become a good business. So before going into the meeting, I would like to tell you that since there is a broad audience of different backgrounds, I would keep it general and try to avoid maximum jargons that people use in terms of business as well as science, so that it is much more simple for everyone to understand. So just introducing how my association with HBNI is. Before my PhD, actually, I joined ACTREC for a master's project under Dr. Prasanna Venkatraman. It goes way back in 2008. So Dr. Prasanna is currently the deputy director of ACTREC. And as introduced, I joined PhD in the same lab, and my thesis was on studying the structure function correlation of proteins involved in protein degradation in human cells. So let us go to what we are talking today. I am part of a small startup called Exact Diagnostic Private Limited, which is currently based, the research facility is currently incubated in Anna University, Chennai, and our office is in Hyderabad. So this startup happened to form because of my association with Professor Shankaran, as you see on the right. I was working as a scientist with Professor Shankaran, where we used to do research on infectious diseases. And apart from the fundamental aspects which we were investigating, we also used to find out targets for diagnostics, and we were trying to develop very cheap and novel diagnostics, which can be potentially a product for the diagnostic industry. So since we happen to do well in that field, we came across Dr. Pavan Asalapuram, who's also an alumni of Anna University and who's currently placed in Sweden, and he owns 2 companies over there. So this work impressed him very much, which led to the formation of our small company called Exact Diagnostics. So initially I was just the only employee, and now currently Jairam has also joined us. So this is a very small team. Professor Shankaran gives his inputs, and people actually, the workforce is just the 2 of us. So let me go to what we are discussing today. So what we are actually discussing today is I want to convince all the PhD students is that to capitalize on what you are learning in the current PhD so that your fundamentals are really strong. And if your fundamentals are strong, the foundation is strong, you can build all skyscrapers. So PhD is very important in this aspect. Of course, PhD does not teach about everything in life, especially you don't learn anything about business when you are taking a science PhD. So there are certain values that need to be added on what you have learned on a PhD, which would eventually help in developing a successful business. So basically, my talk is divided into 4 sections. with very few and simple slides. I would want to convince every one of you, especially the students who are doing PhD, that they are the best candidate for a startup. I shall explain why, and also what is required the extra apart from your what you learn from PhD that you should incorporate in your research and development in your company so that it can become a successful business. And also, in very simple terms, understand what is a business model. Please don't worry about the terminologies. I am not going to use any difficult terminologies because even I am not a management graduate. Therefore, you just need to understand what, in simple terms, what business means and how you can translate your research into a business. So, understanding that in very simple terms. And also, I would, in the end, in a few slides, narrate my experience, what we do in our startup and what are our products. So, coming to the first point, where I would like to convince you people, especially the PhD students, that you are the ideal candidates for a startup. If you see the Harvard Business Review in 2015, you can find that most of the PhD graduates who pass out find that a startup is extremely attractive, more than 85%. This was not, if you take a few years back, this was not the case. Why? Because in an academic setup, you think about academic careers and you are in that environment always. And we are not used to a non-academic career. And therefore, probably that is why there is a skepticism that a startup may not work out. So I would like to stress this point that you are extremely valuable employee because you are a scientist. This is because that it is natural for every PhD student that they are trained to do a lot of research, that is gather information, connect the dots, and they can design a hypothesis based on the study which they do in a much better fashion than compared to those who have not done a PhD. And you can also design the ability to design experiments, test them, analyze them, uh, evaluate them, and communicate. All these are, you spend 5 to 6 years in a PhD program, and a PhD, if you make use of that program very well, and naturally they train you to do so, you will be very good at all these things. It is very well understood. You just need to realize that and apply that in whichever situation you put. The next thing is the two important parts which I feel a PhD student can is better is risk taking, because and handling rejection. You will not find any PhD student in the world who says that all my experiments have been successful. You won't find any principal investigator in the world who say that my paper, every paper I submit to any journal has been accepted. Every person undergoes a rejection, and the ability to face the rejection repeatedly and come out of it, the endurance and perseverance that you find in a PhD student is much higher than a person who has not gone through this program. Maybe there may be a few exceptions, but in a generalized terms, I can vouch for that these qualities will be inherent because of the training that you have taken. Another two important things that a PhD student will always realize at the end of his PhD is that he will always go for the trial and error method, because he knows that at one shot he may get the right answer. So the patience and endurance is naturally developed in a PhD student. And also a PhD, you get a PhD not overnight. You work hard for it, you publish, and you go through so many committees and validate yourself that you are pretty good at it, pretty good at what you're doing. And therefore success is not a sprint, it is a marathon. It is not overnight. And similar is the case of business. Success is not overnight journey. You take any successful businessman, he would have worked hard for at least a decade from scratch to establish his business. And the second thing is that business is not, you have a successful business, that is not success actually. Success comes in maintaining what the business should go on for a long time and it should continue. And maintaining that is even more difficult. And that is what matters actually. So all these qualities are inherent in a PhD. So I hope you will be now convinced that a PhD student you all have these qualities or are developing these qualities. But at the same time, PhD does not teach you a few things which, if you are coming for a business or a startup environment, you may have to inculcate. The academic setup is in such a way that you are given a problem and you investigate thoroughly deep into it and find what is you get and what you don't get, and then end up having a conclusion. But in a startup or an industrial setup, that is not the case. So here, one thing which any person, you may be a very good scientist, you may be very good in designing your experiments, your fundamentals may be excellent, but you should keep one thing in mind if you are going to work in a startup, that is the customer is or the client is the king. He dictates what to do. And therefore, the customer, your idea, your experimental research should always be based on what is the demand of the customer. This is very simple. I am not talking something new, but I am just trying to make you realize that it should be kept in mind constantly when you are working in a startup. Because in a startup, there is not much money, there is not much investment, but at the same time, you have to get the business going. You have a lot of competitors in the market, big players. So you should always think about how the demand is in the market. And what is very important is communication in terms of your idea. So generally, you may think that I have a very novel idea and therefore I should not share, somebody may steal it. I think that is not the right attitude. You need not reveal everything, but at the same time, if you keep communicating to the people, whether it be the customers, your colleagues, or even your friends in the industry, clinicians, or whoever your work is associated, you will find what they require and what the customer requires, based on which you conduct your research in that way, so that it becomes a product which we call the minimum viable product. This product can be sold in the market, but it may not be up to the mark, but at least you should try to bring your product to that level. It can be a product, it can be a technology, but that has to be based on the customer feedback, as well as the demand of what is there in the market. So if you keep these things in mind, uh, you can and you conduct your research in a startup in this way, the chance of survival and generating revenue is good, because ultimately what defines you in a startup is the revenue, how much money you are going to earn in the coming years. So the investment in R&D should be based on this kind of an approach. So the common question that I face from many of the students around my startup is, how do I get an idea and translate it into business? Ideas can be a number of ideas. You may get a novel idea. You may work on an old one. A novel idea is very rare, but it is preferable. But it is very rare. Everybody does not get a path-breaking idea, and then you can validate that idea and create a business. But if you get it, well and good. But as you do your academic research, you work on the same thing by reading the literature, but focus on the problem. How can I improve the product? How can I produce a low cost product? Because that is what gives you an edge in the market. So improvement of the better than the competitors, all these kinds of things should constantly run in a business setup. So so all your R&D is based on these kind of things, these kind of thoughts that should run constantly 24 hours in your mind as you do your research, so that you get a minimum viable product and improve that minimum viable product into a product that can be sold. So the next is what is a very important thing is the money aspect. Uh, if you take academic research, you need funding. You write grants, go present to a government body, and you get the grant. You present preliminary data. It is the similar thing here. That is why a PhD student is used to writing grants with helping the principal investigator and gets to know how to write grants slowly. And this is how you do it in the private sector as well, where there are certain government agencies now like BIRAC and DST, Startup India, which promotes startups. So you can go with your idea, pitch in such a way that with your preliminary data and try to convince the board to fund you. There are private investors as well, but if you don't get both, there are alternative approaches as well. So now I am coming to what we do at Exact, how we dealt with the money issue, because we had money issues when we started. So we didn't have government agencies funding us initially. We didn't have private investors because we didn't have much of preliminary data. We were confident that we could develop a novel diagnostics, we could develop cost-effective diagnostics. But what we went was for alternative approaches. What was this alternative approach? Was you work on your idea, your dream idea, which you want to work on and get the product done. But you need money for it. How do you generate revenue for that? Of course, a small amount of money is invested by the CEO, but that is not enough. So you should actually parallelly generate a business so that you can get things going. So you work as a consultant. That is, you work, use your expertise, solve problems, and then get your client's problem solved, partner with your client, and also do business with your client with the product, or at least get money for the service. So you generate the revenue by working as a consultant and use that money to work on your idea, fund that for your ultimate idea where you are doing, and ultimately develop a product or a technology, and you can use that data to get more funding, or you can even go for the same if the product is very good and competitive and can go into the market. So this is our business model. So you need not follow the, you should not think that this is the right business model. This is our situation. Our situation demanded such an approach. It will differ from product to product, company to company, individual to individual, based on the situation which they are. So now all the business thing is gone. Now we'll go to the science part. And I would just want to let you know that how I went into Exact Diagnostics. So before the company was formed, when I was in the academic thing, I had this problem which every PhD student will have after completing PhD, whether to go into academia or into the industry. But even if you go to the industry, you should be happy there that your academic curiosity is not killed. That was my personal feeling, because I always liked the academic ideas to be translated. So even if I'm in the industry, even in business, I should be doing science. That was my preference personally. So I always preferred a smooth transition to industry. If after PhD or postdoc, or if I'm in the I am an academic scientist. So all this started with the work at Anna University. This was my transition phase before entering into the startup. So what we were doing there was we were actually studying infectious diseases, where we were studying bacteria, different types of pathogens, tuberculosis causing bacteria, typhoid causing, shigellosis, that is dysentery causing, all these kinds of bacteria. We were studying its fundamental aspects of pathogenesis, that is disease causing ability, as well as we were trying to develop detection techniques or diagnostic techniques which are novel or cost effective, so that it can become a product. So we found out that the bacteria secrete certain factors or proteins that are very important for their survival. So our major strategy was to identify unique factors that are secreted by particular bacterium or a pathogen. For example, if I can identify a unique factor secreted by a typhoid causing bacteria, and if I can detect that factor alone in a mixture of so many bacteria, I can say that typhoid causing bacteria is present in that mixture. So using my expertise learned from Prasanna lab in Acton, this is where my PhD comes in play. The fundamentals which I have learned there, using those approaches, we try to exploit and study the factors that are secreted by these bacteria and then exploit it for detection or diagnosis. So our major focus was whatever we detect should be quick. It should not take a long time. It should be cost effective, that is affordable to the public, because there's no point having a big technique, having big machinery. A lot of money needs to be put in. People will not buy it. So if it needs to go to the market, at least it should be affordable comparative to what is existing in the market. And then third thing should be user friendly. That is, any technician, he need not have a very big technical excel. You add this, it should give you the result. So that kind of product is what we were thinking of. So when we were working on salmonella, which is a typhoid causing bacteria, we found that it was secreting a protein called SIIE. It is well known. This is nothing new. But what was our discovery is that we could show that it could catalyze a chemical reaction. It could act as an enzyme, which was the new discovery. So what it could convert a chemical sulfite to sulfate. So it had a sulfite oxidation activity, which is what we discovered. We characterized it thoroughly. We not only characterized it in the organism, outside by recombinant or genetic engineering wise, we proved that this has that catalytic activity. So we published this. I was one of the corresponding authors in this paper, but yes, we have published it. But this was a very important finding, and we wanted to translate it. Can we make this into a diagnostic product? This was our question in Anna University. So this actually kindled us into probing further, and we developed a solution, a reagent, which is pink in color. And our method of detection was very simple. You take the sample, which is stool sample, fecal sample, which is normally, you get dysentery during typhoid. So you take the sample, you inoculate in a media, allow the bacteria to grow. All kinds of bacteria grow. If salmonella or the typhoid bacteria is there, it will also grow. So grow it for 6 hours and then add this pink solution. And if you add the pink solution, if you don't have salmonella, it is pink in color. And if you are infected with the typhoid causing bacteria, it becomes colorless. This was our design of what we discovered. The product was this reagent. You add, visually you can distinguish whether it is salmonella positive or salmonella negative. It was very quick. And you can see that if we found that only salmonella this happens, it doesn't happen for other organisms. So it is specific for typhoid causing organism. So, so this actually, if you compare the price as well as the time taken of the current methods that are used for detecting typhoid, the most common method is Widal test. So the Widal test is not an antibody-based test for the non-biology students. You will now know what antibody is because of COVID. So your antibodies are generated only after some time after infection. So if you get typhoid today, tomorrow you go and test, you will be actually negative because enough antibodies won't be there in your bloodstream. Therefore, you have to go at the 7th day or the 8th day to confirm whether you have a typhoid. So, and it is also very expensive. The culture method is that the normal microbiology method to identify the typhoid organism is also there, but that also costs around 80 to 150 depending on the lab and place, city. But our method, you can see, directly identifies salmonella. And it takes only 6 hours, and we can price it below 50. So in that way, our product satisfies everything. Rapid, 6 hours, cost effective compared to what is existing, it is cost effective, and it is user friendly. You just have to put it in a media, add your reagent. Making the reagent is also very easy. So all these things show that we could develop diagnostic in a very cheap and a quick approach that can become a potential product. So this is what this product could be used in the diagnostic industry for typhoid detection. It can also be used in the food industry where you find typhoid bacterial contamination, and that those products should be avoided. So for food testing also this can be used. So now we were very confident that we can make some kind of diagnostic for multiple organisms. And that is when we happened to meet Dr. Pavan Asalapuram in Sweden. And then everything turned out to a startup environment. And Anna University helped us in incubating the company in Anna University itself. And that is how Exact Diagnotech in 2019 was formed and was incubated in Anna University. So this is where our startup story begins. But now we didn't have enough money to do all kinds of. Anna University had a lot of money. We had a grant from DBT as well as DST. So everything was going very smoothly. Now we needed to partner with somebody and work as a consultant. As I said, that was our business model. And we partnered with a company in Sweden called Apires. It was also a small company which produced an enzyme called Apires, or its commercial name is called AP1, which is a patented enzyme. So we had an agreement, and they gave, and this actually converts nucleotide triphosphates to nucleotide monophosphates. That is what AP1 does. So what are these biology students will know. So the non-biology students don't bother. So normally Apires is used in sequencing, which is related to DNA sequencing. So now in Sweden, they wanted to use this enzyme in clinical samples, especially urine. So when they used this AP1 in urine, it was not working. And they did multiple tests, many things, but things were not working. And they wanted us to find, this is what our problem was. They were saying that the salts in the urine, the magnesium or calcium, all these may affect the enzyme, so mutate the enzyme. You find out a solution. So Exact Diagnotech had to find out the solution. This was our first make or break project to make AP1 active in clinical samples. So we actually worked on AP1. And to just introduce you to enzymology, enzyme is a catalyst, which is a protein that converts substrate to product. So most of you will be knowing that. So what Apires was suggesting us, and they were understanding, was the components of urine were affecting AP1, the enzyme. The enzyme was affected by the urine, and therefore enzyme was inactive, so it was not working. So what we found was something different. So that was another thing which I want to say to all of you is that there are many things may appear so complex, but we may neglect certain simple details. So when we were carefully observing our experiment, we found out that the urine components were not inhibiting the enzyme. It was inhibiting the substrate, and it was precipitating it, so the reaction was not happening. Anyway, the problem exists, but we identified the problem. When we identify the problem, there is a possibility of a solution. So once we developed a method. Now I cannot reveal the details of the method, but we developed a method wherein the AP1 can be resistant in a very high salt solution. Forget urine. Urine, so if you put it urine, it will not work. But when we developed a method in such a way that you can see 88% of the activity is retained, is brought back. And we also put it in a high salt solution like seawater. and we still made it work. So that shows how confident we are that this enzyme can potentially work after we adopt this method which we discovered. So our new cost effective, it is very cheap also. We didn't, use very expensive reagents, but we could retain the activity in clinical samples. So this was our make or break project. Applidace was very happy with these results and continued to partner with us. But I didn't stop working with Applidace because my curiosity, academic curiosity kept going, and I continued to work on Appli1 by mutating. This is where the coaching or training from Prasanna lab helped me, especially looking at structures of protein and identifying residues, changing, making new variants of enzymes and see what happens. So I had this gut feeling that API1, which is a pyrophosphatase, as you can see in the title, it can only act on pyrophosphates, a class of molecules having two phosphates. It can never act on single phosphate molecule. But I felt that if you can modify based on the structure, which I could study based on the literature and everything, I could mutate in such a way that it can be converted into an organic phosphate. And therefore, I discovered certain mutations that we performed in such a way that we could enhance the substrates, mean more substrates for API1. That is, it became an organic phosphate as well as a pyrophosphates. So this new version of API1 was patented. So what we did, we converted API1 to another protein called API2, which is an organic phosphatase as well as a pyrophosphatase. So this was, we have a Swedish patent now along with Apirase, where we can use it for more applications, especially in the biochemical as well as immunoassay. So what is the advantage of this product, API2? It is low cost. It is used as it can be used as a pyrophosphatase and an phosphatase. It is resistant to agents like EDTA. It requires no cofactor, and it can even work in water. You put it in water, it will work. So the new substrates which we discovered are very common substrates which are used in detection platforms. Now, what are the competitors? Acid phosphatase and alkaline phosphatase. Most of the life sciences students will know this because they will be using it in the lab. Now, these require cofactors. These are inhibited by EDTA. If you have anything, a system with EDTA, you cannot work with these enzymes. But API2 can work. And it can even work in physiological pH. That is 7 to 7.58. It can work very nicely. So you see now, what I wanted to stress now is that we have enhanced the ability or the usage of this product with another application. So we still worked on API1, and the paper gave me a clue that it can be used for another application, that is biofilms. Bacteria grow in areas attached to the surface, and they grow profusely if they are not cleaned well. And then they form biofilms, a slimy layer, which ultimately leads to, you can even be resistant to antibiotic. So if you have a oral unhygienic environment you don't brush properly, you eat all kinds of sweets, and the oral hygiene is not maintained, you are bound to get biofilms on your oral surface and get gum disorders or dental plaques or cavities. So this, so what I studied was the triphosphates, were the signaling molecules that were helping biofilm maturation, biofilm growth, especially in the oral region. So I thought that if I can use API1 that can destroy the signal, can I inhibit biofilm and have healthy teeth? This was our hypothesis. So that excited me in such a way, but you cannot go and put API1 in the mouth and say, oh, this is now going. So that is where certain prerequisites have to be fulfilled. So I should be able to put API1 in a mouthwash or a toothpaste. Now this is where the application comes and where the PhD student needs to focus. And it should be miscible in an organic solvent because all these mouthwashes and toothpaste are organic in nature, hydrophobic in nature. And it should be stable in room temperature. I cannot keep my mouthwash in fridge. People won't use it. It should be in room temperature. In the medical shop, it should be in the room temperature. can it attach to my teeth so that the enzyme remains for some time and then it can clear the biofilm. So I had to engineer the AP1 in such a way that I added a lipid tag to it and made it a lipid modified AP1, which is called ALM, which we named for currently ALM. And then we mixed with mouthwash and saw. You can find that AP1 LM was the lipid modified version was much more stable in the organic solvent, and for a number of days we are still testing for more days, but at least 30 days data I'm giving you, and it can other much efficiently on a surface, a plastic surface, even table also it will other. So we could test its. So all these criteria, ability to mix in mouthwash, it was stable in room temperature, it could attach on a surface, it could be miscible in an organic solvent. Once we were sure that all these criteria were met, we wanted to go for the major test. Does it inhibit biofilm? So that was the next aspect, where we took different bacteria, and especially tough bacteria, which forms very tough biofilms. And once you can see that addition, we don't add any disinfectant, only the enzyme. Addition of only the enzyme, that is AP1 lipid modified, you find that the biofilms tends to disappear. So the adherence of the bacteria, it doesn't kill the bacteria, but it prevents the adhesion of the bacteria on the surface. And it because this kills the signaling molecule for adhesion. And that is how we actually presented to a head of pediatric and preventive dentistry in Sri Ramachandra University, Dr. Muthu, who himself is an entrepreneur and is owning a startup. And he was very impressed with these results, and he told us that this he would help us in testing it in dental caries model in his lab, and if possible, the clinical trials. And he told us that only solution for biofilms in the teeth today is fluoride, where they use fluoridated toothpaste. But many people are against the usage of fluoride. So therefore, any alternative would be very impressive. So LM could be a possible solution. Where we have to wait and see. So this is just the beginning. So what I wanted to tell all of you, I just going is a callback where I had mentioned a minimal value viable product. So that was AP1 for us, the first project. But you can improve that product in such a way that we engineered it for a clinical studies. We engineered it to make it a different kind of an enzyme so that it can be used for different platforms, biochemical and immunoassay platform, and you can use it for dental application. So one product, multiple application. This is actually a boon. And all may not work, all may not hit the market, all may not go and make you money, but at least one will do. That is what I would like to tell you, that get that minimal viable product, work on it in such a way, keeping in mind what is the demand in the society, what the demand in the market. That's what will generate the revenue. So that after that, apart from this AP1 business, we also work on certain ornamental enzymes. I call these enzymes ornamental or proteins ornamental because they are highly expensive and they are not manufactured in India. Chinese manufacture it, US and European countries manufacture it. So we were thinking of bringing down the price, but it is very difficult because these enzymes, challenging enzymes to purify. And current market price is 20 rupees. We are trying to bring it down to 4 or even less than 1. That is my aim actually. So that, so if anybody, life science students want these enzymes, thermostable ligase, 1 hour, 55 degree, all your cloning will work. You can contact me. I will give you a free sample, a marketing strategy through my presentation. So you can give. So I'm almost into 40 minutes. So I will go to the long-term plans of my company. Our dream project is to develop a diagnostic where I can have a simple, cost-effective diagnostic for every pathogen. That because most clinicians today, you can say this infection is there, that infection. They want to know what pathogen is there. That is what every infectious disease clinician tells us. So if you are able to give us a cheap pathogen detection method, four pathogen set like salmonella, shigella, dysentery related pathogens in one kit, pseudomonas pathogen in another kit. So if that is possible in a very cost effective and quick way, we are working on that lines. So the second thing which we was based on my friend's suggestion, he is working in a clinical setup. So I was telling him that we could actually count exactly how much bacteria is there without much investment. People use bacterial counters, which for urinary infection to count bacteria in millions. So he told me, why don't you develop that product, because that costs lakhs, where you can sell your small kit for a few thousands. So we are working on that also. And also we are working on a platform technology to make sensitive antibodies. That is, antibodies that have very high specificity. Normally, you dilute antibody 1 in 1,000, 1 in 2,000. We are thinking of using antibody for 1 in 50,000, 1 in 20. So much sensitive. So if we can develop a technology, and in collaboration, we could actually cover a much more amount of applications and locally supply it to all the clinical laboratories around us and get some business going. So this is what our startup is doing in the planning in the long term. So I will conclude my talk with these concluding statements just to remind you that PhD is the tool for a startup, and it keeps your academic curiosity. The startup, you can keep your academic curiosity going if you are really interested in continuing to do science. If you go to a very established industry, you will have to listen to what they say. And that is how here you can do both. If things work according to the product, you go with the product. And you keep doing multiple things. Some things go into academic line actually and doesn't eventually end as a product. You publish it. It will add to your CV. So you keep your academic curiosity going on one side. You are running the show. And if you are able to satisfy your CEO by giving some products at the same time doing your own research, it may go above 30 or sometimes even late 30s, depending on your situation. It is still early. You can take risks. And if you succeed, great. If not, who cares? You can go and move on. So if you want to try different things, it is just right now. This is the time you can try different things. And one thing you should always remember is that there won't be man. I worked for 1.5 years alone, all alone in the incubation center. Nobody in my company assisted. I was the only employee. So therefore, you have to do multiple roles. Because now I have an assistant. The manpower keeps increasing as you start developing business, and your confidence in the CEO also goes high. So you have to do every role. The technician's role, the scientific assistant's role, the accountant's role, the ordering, writing the grant, performing the experiment, designing the experiment, planning the experiment, everything. If it fails, you have to attend the meeting, you have to organize meetings with the other collaborators, everything done only with few people. So you will learn multiple things at this young age. And the growth outside it may sound very, to look at it, there's nothing actually, but the growth is exponential, and eventually the fruits will come. And don't look at the fruits right now, just enjoy what we are doing. If it succeeds, fine. If it does not, if you're still good at it, people will pick you. So that you please keep in mind. And as I said, a novel idea, high confidence, you focus on it and finish the idea to bring it to the market. If you don't have a novel idea, don't worry, multitask as we did multiple projects. If you do 10, 2 will go through. And with those 2, even if one goes through, your company will be happy, and that revenue is enough for your company to make profit. And also remember that you should have a good business model, which varies from person to person, company to company, product to product. So I don't have a very big story to tell because it's only 2 years since I have been in the startup. But we are crawling. We are not lying down, but we have started to crawl. and I'm sure that we will walk one day briskly, and I hope we will do that soon. We are still hoping, although we have had good businesses with partners, but it would be our dream that when Exact Diagnostic has its own product, and when we sell that, it will be a big thing for us. So I would like to thank everybody for giving me a patient hearing. I would be happy to take questions, but before that, I would like to thank a few people without whom all this wouldn't have happened. Exact Diagnostic, of course, with my CEO and Professor Shankaran, who's the advisor, and my research assistant, Jairam. Pawan has been very patient and kind CEO and helping us, without putting much pressure and teaching me what business is. I was completely naive in this, and whatever I'm speaking about business is based on the inputs or what Pawan has taught me, and also Professor Shankar, who has given me all his knowledge regarding the infectious disease part, and also his lab, and also allowing us to incubate in Anna University. Of course, all other important faculties of Anna University who helped us, go through all the initial tough period and try to establish initially. And then, of course, ACTREC and HBNI, without whom all my fundamental knowledge would not be there. And especially the Prasanna Lab family, ma'am, and all the lab members who have been, played a major role, in fact, 6 years in ACTREC has been a wonderful experience, a learning experience, and it is the fundamental knowledge gained in that lab that is helping me to do what I'm doing today. And I should not forget the role all the deputy directors, former deputy directors, the directors and former directors played, all the other faculty, the student community of ACTREC, who have played a major role in shaping every student, including me, to what we are today. So I thank every one of you for this patient hearing, and I would be happy to take questions if there are any. Thank you very much.
Thank you very much. Thank you very much, Dr. Nikhil Sangith. Really, HBNI is proud of you, pride of our former student to take up this challenge and really started brisk walking. Looks you have started that. Okay, over to Rucha Kulkarni. If any question is there from ACTREC or anybody else, you can put up there, or you can take over. Rucha.
Thank you, Dr. Nikhil Sangith, for a very informative and a very interesting talk. I would request Mr. Anurag from HBNI to check if there are any questions from the YouTube comment section, as we will not be able to see those over here. Is Mr. Anurag connected with us?
Okay, our Vice Chancellor has one question to Dr. Sangith.
Yeah, please.
Very good afternoon, Dr. Sangith. Before I ask the question, first of all, let me convey our sincere appreciation to you and your work. And I am sure that with the kind of approach you have outlined, you will not only start walking briskly, we hope you will soon run fast and become a winner. And we also want to thank Dr. Prasanna Venkatraman for creating such students who have who are also brave to go into the world and succeed. Okay, having said that, my question is from more HBNI centric and student centric. You said that the Anna University provided an environment for you to grow.
Yes.
You are alumni of HBNI. Now, can you tell me, if HBNI has to provide such an environment to grow, what should it do? What did you get in Anna University, which has to be created at HBNI?
Anna University is one of the centers for University Innovation Cluster, which is organized by the Government of India. The University Innovation Cluster, there are 18 centers in India, out of which Anna University is one of them. So in the university innovation cluster, they encourage innovation and recruit PhD students with the innovative projects, fund them for a period of 3 years. And if they are good at developing a product, they push them towards the related industry. So that program actually was, intersecting with our lab also. So I was also connected with the University Innovation Cluster program because Professor Shankarvan was the coordinator of that also. So that gave me an environment to do that. Innovation cluster was present, which encouraged exclusively only innovation. So they used to recruit PhD students and BTech students, M students, give them a fellowship and grant also, which was funded by Government of India. So for a period of 3 years. So which will be reviewed every year. So that innovation cluster led to an incubation center. So many companies started to incubate. So that environment started coming up in the Anna University campus.
Second is more of a hope and suggestion than a question.
Sure.
You mentioned that you are still a very, very small company and you have just added one research person to your team. And I'm sure as you go along, you are going to find many more new products that you can make and succeed.
Yes.
I only hope that through this program and maybe subsequently, I suggest that if you agree, we could have a meeting of alumni of HBNI and specifically those working in those who are interested in entrepreneurship, our PhD students in last few years.
Sure.
I hope some of them or one of them or two of them will be able to join you if required or take up similar paths by such more closer interaction, because this is not usually possible in a colloquium or a webinar like this. It takes some time to, just for the idea to sink. But maybe after a few days, some student will start thinking, why not I also do this? So we would like to follow it up with a meeting with, let's say, students who are entrepreneurship minded. I am having a meeting shortly with the Research Scholars Forum, and I will suggest to them that they could invite you and you could have an informal discussion with them to give them some ideas like this.
I would be happy to. Sure, sir. Anytime you can let me know. We will meet.
Professor Prasanna, you have something to say?
Yes. Thank you. And I am seeing Nikhil after so many years. And, this happens to students at their defense, that they become emotional. So this emotion is not because he is showing a success path. I've always tried to build these qualities in my students to be able to pursue what they want, pursue with confidence and bravery, and not worry about what is the immediate success, and to be able to love what they do. Today, he just demonstrated to me, and I always keep worrying whether I did the right thing, have I done the right thing? If you look at all my students, they may not have gotten fantastic publications like in high impact journals, but they're all doing very well in each of the hemisphere that they have taken. I have only few students because the career started late. 5 of my students have graduated in different places. One is with me as a postdoctoral fellow. These qualities, you think that you should imbibe in them, and they are learning it. And it is when it fructifies in this manner. And Nikhil has always been a very lucid speaker, very clear in his mind. And when he communicates what is in his mind. He did it again, and in a completely different position, keeping that academic curiosity, but also bringing up the industry flavor and to be able to understand what is the need of this country. I am deeply moved, and I am very proud today to have listened to Nikhil, and I'm very happy, and it brings me immense happiness that I'm doing the right thing to my students. I'm not talking about the success. He will run, he will achieve many things, but there are some qualities that I wanted to see in my students that is lifelong. It has just nothing to do with their PhD or post PhD or industry. There are some qualities that I wanted in my students. And first thing is that research and pursuing new aims should be with the boldness, that it should be bindas. The idea should be pursued with a bindas mind. I keep saying that. And he just proved to me that it is possible. And I enjoyed your talk, Nikhil. I'm very, very
Thank you so
happy.
much. Very honored.
Congratulations, and let the years will make sure that you will become more and more successful, and also happy in continuing your research pursuit. Thank you, and thank you HBNI and Professor Rao, Professor Maiti, Professor Nayak for giving him this opportunity and making me listen to this. I'm very happy today. Thank you very much.
Thank you. Thank you so much.
Thank you. Thank you. Rucha, you have any question as a student or anybody, any of your friends have any question? One chat mode, there is a question.
Yeah, in the chat box, there's one question. I guess it's from HBNI. It's for Dr. Nikhil. What's your outlook regarding lab-on-chip based diagnostic devices market in India? So we wanted to know what you think about this lab-on-chip based diagnostic devices.
Yes, it sounds very good, but when we have to come down to the ground to understand whether these technologies are feasible. So, lab on chip, what do you mean? For example, okay, he says that cheaper blood glucometric test, point of care device, definitely it is possible. So it is based on everything which you look at. So in the case of cheaper glucometric test, of course, now there are lots in the market. You can get small strips which this is electrochemical based. So you will have to, so if you should find some other problem, because blood glucometric tests have become very common, and you can get so many instruments in the local medical shop. So if you want, you have to find out the problem that the lab on chip can be made, but it should be difficult to do, a challenging aspect. Either it is very expensive, or so that you bring down the cost, which is actually works in countries like India, and also if you have a novel technology, then go for it. No problem. But definitely it is possible. It is a case-to-case difference, and definitely it is possible. Have I answered the question?
Do you have, Mr. Anurag, do you have any question? No question. Okay. In that case, Rucha, you can close the meeting by usual way, thanking the speaker and all. Rucha, you are there?
Yeah, I would. Yeah, I'm there. Yeah, so I hope there are no more questions to be discussed from ACTREC as well. I mean, there's no put up over here. So I would like to thank our speaker, Dr. Nikhil Sangith, for his very informative and a very a talk which was on a totally different topic that we don't come across in our day-to-day lives actually. So we keep on listening to talks on scientific topics, but then this was something really different, really interesting, and yeah, I mean it took us through a very very exciting journey, I would say, and we would surely like to explore this part as well after we are done with our PhD. As you rightly said, that doing PhD is very important, so because it makes you more and more curious to know and to explore things, and that curiosity should stay throughout, even if you are not into academics and you just bridge this gap between academics and industry. So that should stay with us throughout. So that is something that I thought is very true and really should think about it. So thank you, Dr. Nikhil, once again, and
Thank,
from the entire student community of FactTrek.
thank you.
And
Thank
we
you
all
so
wish you all the best.
Thank you so much. It's been an honor. Thank you. Thank you so much.
Okay, we close here. Thank you, Rucha, for conducting nicely, and of
Yeah.
course, thank you, Dr. Nikhil Sangith.
Thank you.
We
Thank
hope
you, sir. Thank you, sir.
we'll be talking
Sure, sure.
very soon again.
Sure, sir.
Okay, because the
Looking
HBNI is
forward.
thinking to make certain in a similar way some incubation center, very preliminary stage at the thought process stage. So
Sure.
probably we need help from you and similar people. We are working on that.
Yes.
That's what our Professor, Vice Chancellor Professor Rao was talking about you. Okay, thank
Sure.
you then. We close
Thank
here.
you so
We
much.
close the
Thank
meeting
you.
for today. Thank you. Thank you very much.
Thank you, sir. Thank you all.
Bye. Thank you. Thank you so much.